Afkhami here to talk about his experience through his studies in immunology. Please take note if you have any questions throughout the presentation. We'll have a segment at the end where you can ask questions. I'll pass it over to him. Hi, Sam.
Hey, how's it going? How's everyone?
Good.
Good. So what's the format for this? Should I just go straight into my presentation, or if you guys have questions for me before I start, whatever you guys want.
We can jump right into the presentation.
Yeah? Okay, sure. Well, thanks for having me. It's a great initiative you guys have. And
thanks for being here.
Yeah, no problem. Can you guys see my screen okay?
Yep.
So today, I guess I'll give a little bit of a brief presentation on what we've been doing in terms of, what is immunology, how is it applied across different disciplines in terms of medicine and discovery, and then I'll focus a bit on what I've been doing in terms of developing vaccines for different things such as tuberculosis and our recent initiative at MAC in developing homegrown COVID-19 vaccines. So a little bit about myself. So right now I'm doing my postdoc at McMaster in the Department of Health Sciences at the McMaster Immunology Research Centre. Originally, when I started my postdoc, I was working on a couple of clinical trials involving vaccines against tuberculosis. But as this pandemic started to emerge, McMaster had set up this initiative known as the Global Nexus for Pandemics and Biological Threats. So we've been working on leading a couple of different vaccine strategies in developing these sort of next generation vectors for COVID-19. But really, before I got to this point, I originally did my bachelor's at MAC as well. I've been pretty much homegrown at MAC for the last God knows how many years in the Honours Bio program. And throughout my entire Honours Biology degree, I really didn't know what I wanted to do. And it really wasn't until our third year where we took an introductory to immunology course where I found that really my passion laid in immunology and things related to immunology itself. So from there, I did a undergraduate thesis in a cancer biology lab here with Jonathan Bramson, who studies different therapies for cancer. And, that was exciting and all, but at the same time, it was also a lot of unknowns of what I wanted to do from there. I knew I liked the research, but didn't know really where I wanted to go from there. And eventually I decided to do grad studies in immunology, focusing more on vaccine development. And from there, I essentially transitioned into where I'm at now, which is my postdoc, once again, focusing on vaccine design and development. So the lab that I'm part of is the Zing Lab. So the lab that I joined originally, the real goal of the work that we do is understanding not only respiratory immunology, but really understanding how pathogens interact with the immune system and how we can use that understanding to develop new vaccines and therapeutics against a range of respiratory pathogens. So what I'll present today is a little bit of a brief history of immunology, which I think is always nice to touch upon, where the roots are of the work that we do, what immunology now is in modern times, the work we've done in developing vaccines against TB, and our sort of forefront efforts against COVID-19. So when I first started learning about immunology, people always considered it as a branch of biology, where we essentially was the study of the immune system. But I think nowadays, I think it's better to challenge this idea in the sense that immunology itself really sits at the core of a variety of different disciplines. So our understandings of infectious diseases, vaccines, and therapeutics, as well as things such as cancer therapy and diagnostics have really stemmed from immunology itself. And the field itself has been it's rather new. It's only a couple of centuries old, but if we look further back into humanity's history, it's actually been very interesting to see that immunology and concepts of the immune system were actually actively applied by humanity to better itself before we even knew what it was. And I think one of the best examples of this is dating back to when smallpox was essentially ravaging the world. So smallpox is a viral infection that is essentially associated with these development of these sort of pustules on the skin. And when we look back into documents as early as the 16th century, the these were records from China, which suggested that people were actually taking these pustules off of people who were originally infected and essentially sniffing them as a way of, they noticed that when you do this, it actually protects them from getting smallpox. And this itself was really the earliest form of vaccination without even knowing what we were doing. And this sort of idea of taking these pustules and inhaling them to provide yourself immunity carried on for multiple centuries after that. And really, it wasn't until, I would say, about the 18th century when a medical student at the time, which I think a lot of people have heard about, Edward Jenner, started seeing certain trends among, in particular, milkmaids. So he saw that milkmaids, for some reason, didn't get smallpox. And when he looked at these individuals closer, they saw that they had sores on their hands that were similar to smallpox, but they were actually the cow form of smallpox, which was known as cowpox. So cowpox didn't cause bad disease in humans like the smallpox variant did. So he actually carried out some of the earlier experiments to show that if you expose someone to a certain agent, they're actually protected against that agent. And he essentially coined the term vaccination, which came from variolae vaccinae, which essentially means smallpox of the cow. So the word vaccine had actually originated from cow. But even when these were identified, no one knew what immunology still was. They just now had this sort of experimental understanding that if you're exposed to something, you're protected from something. And it wasn't until 2 centuries later where Eli Mechkanov and Paul Enrich really provided the real foundations of what we know as immunology today. So Mechkanov had a microscope, and he was able to see that when you cause damage to an organism, there's all these little bacteria get in, and then all these cells from that organism seem to cloud around that bug. And Enrich was able to find that there's something within the blood that seems to prevent an infection from occurring after you've been initially exposed to it. So their findings essentially provided the basis of what we understand today as cellular immunity, which are the cells of our immune system, and what we consider humoral immunity, which are antibodies. So humor comes from like serum. So these guys were really essentially the foundation of what immunology is today. And if we look at current modern times, we actually define our immune system by 2 major pillars. We call it the innate immune system and our adaptive immune system. So the innate immune system is really that frontline barrier to infection that we have. So our skin, for example, is one of the largest organs and largest immune organs that we have. The innate system is rapid, provides really immediate to a couple of hours of immediate response to an infection, and is hardwired within our genes. Whereas our adaptive response takes time to develop, but it's a lot more specific, it's a lot more targeted. And that's what really has memory, that we can, when we're exposed to something, our immune system can recognize what happens later on. So this understanding of the basis of immunity has really given rise to a lot of discoveries, especially in the recent years. So I talk about it being at the core of a variety of disciplines. And really, the fact is that immunology does sit at the core of a lot of the things that we study today, and a lot of the medical achievements that we've had have really come from our understanding of the immune system. So a lot of the focus of what I'll present today is on vaccines. So obviously, as I've discussed in a couple of previous slides, we know that vaccines are given in a way to prevent infection or a disease. And if we look at the history of vaccines among humanity, we've seen that there's been a lot of candidates that have essentially completely eradicated certain diseases among us. So smallpox was an example that I've given, but we've seen that vaccination has reduced the rates of a variety of things like measles, mumps, rubella. But there's still many pathogens out there that it's just been notoriously difficult to vaccinate against. And some of the most classical ones that we've focused on in immunology is malaria and HIV, which in turn really contribute to hundreds of thousands of deaths, and tuberculosis, which we know on a yearly kills over 1 million people. So that's where it really puts me into describing what I've done throughout my PhD initially, which has been really the journey in TB vaccine development. So TB itself is this bacterial pathogen that's primarily spread between individuals through coughing, sort of aerosol droplets, and it's primarily an infection of the lungs. And the way you can think about TB is that when this bacteria gets within your lungs, your immune system tries to fight it, but it's technically unable to control it in a lot of cases. And essentially it walls that bacteria off. But the issue with that is that these bacteria continuously can evolve and grow and replicate, eventually leading to destroying a lot of the tissues within the lungs, such as the vasculature and the airways, and essentially people die from their lungs being destroyed from the bacteria. So with that being said, there's been a lot of advancements in how we treat TB. So things beyond our understanding of medicine, things like proper nutrition and sanitation have had major impacts. And I think that's something I really want to focus as well, is that medicine isn't always the direct answer for dealing with up and coming infectious diseases. And when we look in places like in Europe, the mortality seen with TB has continuously dropped as we've advanced our understanding of sanitation, hygiene, and education. But if we look at certain parts of the world like South Africa and India, TB still remains one of the major causes of death within these places. So even though we've had all these advancements, mortality rates of these diseases still remains incredibly high, as much as it used to over 10 to 20 years ago. So this really speaks to the shortcomings of the therapeutics that we have. A handful of people here have probably heard of BCG. So BCG was one of the first vaccines ever designed to provide immunity against tuberculosis. So it's been rather indispensable in protecting against childhood tuberculosis, but the more we've studied this vaccine, the more we begin to understand that it doesn't provide protection against the pulmonary form of the disease, which is the most common one. So it really speaks to the shortcomings of this vaccine. So there's been a real need for developing newer strategies, newer vaccination strategies in this fight against ending tuberculosis. So the question that really came up in my PhD is, how do we improve vaccine formulations? And in theory, it's simple, right? We find a target, we incorporate it into a well-known vaccine platform and apply it as a vaccine itself. But BCG itself essentially was this, and it wasn't perfect. So what are we really missing here? And this really comes and speaks to the idea of modern immunology and modern vaccinology, where we have to really appreciate that pathogens are smart and they've evolved with us for centuries. So if we're going to find ways to vaccinate against these very tricky bugs, we have to find innovative ways to combat them. So one of the biggest things that I think is becoming more and more apparent when it comes to developing vaccines and therapeutics is we need to really understand the life cycle of pathogens and how they interact with our immune system. So TB, as I said, transmits between individuals in these aerosol droplets. And when people are exposed, they develop active disease. And when people develop active disease, they go out and get antibiotic therapy. So antibiotics have been really the staple in curing disease. The issue with that is that antibiotics aren't perfect and people don't always follow their regimens properly. So what happens is that people who might think that they're cured after taking a couple of pills actually fall back into this sort of state of active disease and go back to transmitting it to other individuals around them. So we can stop this by developing prophylactic vaccines, so vaccines that are given before infection, but we know that that hasn't worked with things like BCG. So why not develop therapeutic interventions that can aid or help antibiotics carry out their job? So antibiotics have remained the gold standard for decades now for treating tuberculosis. But the issue with these antibiotics is that they take a long time to work. So unlike certain bacterial infections that we get that you need antibiotics for 1 or 2 weeks, TB requires a minimum of 6 months of daily antibiotic taking to even have a chance of treating the disease. And the issue with that is drug resistance, and we talk about antibiotic resistance, has continuously been on the rise. And for something like TB, where you go from a minimum of 6 months of therapy, when you start developing resistance against these antibiotics, some individuals have to take antibiotics from 2 to 4 years daily in their life to even have a chance of treating the disease. But the issue with that is when treatments are long, it's hard to have people stick to them. So we consider individuals taking their drugs versus not taking their drugs, a concept known as patient adherence. So how well does someone adhere to treatment? So there's a lot of factors that lead to people having fragmentation in their adherence to antibiotics. So in this example here, if someone presents to a clinic with an active disease, they're given antibiotics and they start taking the antibiotics, but there's a lot of factors that lead to them not finishing their therapy for the required time. So when you have incomplete therapy or this fragmented adherence, what happens is not only do you have relapse of active disease, but you're actually developing a perfect environment to develop antibacterial or antibiotic resistance. So this poor adherence to antibiotics is associated with not only that disease relapse that I talked about, but drug resistance. So we know that the immune system is incapable of clearing infection, and we know that antibiotics are hindered by the length of therapy and the complexity needed to treat disease. So can we develop a vaccine that can address the issues of a poor immune system and also address the issues of the antibiotic therapy length? So within our lab, one of our objectives was to really, understand what happens during an infection that allows MTB to be so resistant to antibiotics and be able to bypass the host immune system. And what we've done is essentially we've isolated these factors that we consider resistant factors to the bug and used them in generation of next generation TB vaccines. And I won't go into too much detail about it, but we've used these resistance factors to generate virus vectored vaccines against TB. So essentially the strategy that we've successfully implemented is that in combination with antibiotics, we actually deliver vaccines directly into the lungs. So as an adjunct therapy or in combination with antibiotics. And in doing so, our goal with these vaccinations is to sort of boost that host immune response that initially was failing against controlling TB. And in turn of boosting these responses, not only can we accelerate the clearance of bacteria, but help reduce disease severity and in turn accelerate disease treatment. So unlike what we normally hear in the news about vaccines being given to prevent an infection, we've been working on developing vaccines that can be given during active infection and aid in the resolution of disease. So one of the major takeaways that we had worked on was that we can technically develop strategies to immunize individuals to enhance not only protect them from an infection, but also be working alongside other issues we have with treating that disease. So in this case, was the duration of antibiotic therapy. So developing new vaccines really requires an intimate understanding and really a prediction of a pathogen's life cycle. And I think that's a major concern when we talk about this sort of news of COVID-19 vaccines. So when we look in the news and we see all these different vaccines that are coming out and getting approved, they've shown phenomenal efficacy. There's no doubt about that. And we consider these as first generation vaccines. So these vaccines essentially all target the same thing on the SARS-CoV-2 virus. We call this the spike protein. It's on the surface of these viruses. And essentially, these vaccines are designed to induce neutralizing antibodies that can bind to these spike proteins and prevent infection. But these emerging variants that we hear more and more about in the news have beginning to pose a serious threat to the efficacy of the vaccines we have. And that's because as spike mutates, the immune responses we generate with these vaccines against that original spike may not be as effective. So these variants emerge naturally because viruses mutate, but also from immune pressures, vaccine pressures, and whatnot. So we really need to develop strategies to combat these emerging variants. So one way is we can continuously modify our vaccines, but it really kind of feels like an arms race. So Moderna, for example, began testing a new version of their vaccine to target some of the emerging variants. But although this strategy does work, I think there needs to be better vaccines developed that can cast a broader immunological net. So a vaccine that can provide pan immunity against any potential variants that emerge. And like I've talked about within this presentation, the life cycle of a pathogen and how it interacts with the host really holds the keys of developing these next generation vaccines. And one thing that we've been working on here at MAC is developing these sort of vaccines that can provide pan-variant immunity based on our understanding of what these viruses do, how they mutate, what they mutate versus what they don't mutate, and using those in this next generation of vaccines that we're constructing. So one additional thing that we're also doing is that unlike a lot of the vaccines that we hear about to date, which are given as a jab in the arm, we've developed these strategies to provide the vaccine directly into the lungs. So we know that that's where the portal of infection is for coronaviruses. So if we can drive vaccine responses right at that site of infection, we might have a better chance of preventing any potential variants from emerging from, for example, incomplete immunity. So getting out of the pandemic is the first step, and that's exactly what these first generation vaccines are going to do. But preventing the next one requires us a better understanding on how to develop broader and more pan-variant vaccine strategies. Yeah, and if, I'd be happy to take any questions about the presentation or anything you guys want to talk about.
We have a question in the chat from Ryan. He's asking, how are vaccines directly applied to the lungs?
That's a good question. So the technology is very similar to puffers. So essentially it's a puffer-based technology where the vaccine's loaded into like an aerosolizing unit. You essentially press it and you breathe it in. I'm happy to answer questions about grad school too if you have or anything.
Well, I have a question if no one else does. What's the one tip you'd give anyone wanting to go into graduate studies?
What, can you ask that again?
Like the number one tip you'd give a student if they're looking to go into graduate studies?
Oh yeah, so it depends if you want to do dry lab or wet lab research. So the work that we do in the lab here is wet lab because we have direct hands-on work with experiments. Some people like to do things like epidemiology, or they want to do, for example, programming and bioinformatics. It really comes down to what your end goal is, first of all, as a career. So for me, I knew I wanted to go into industry down the road. And if you want to do industrial research at like big pharmaceutical companies, you have to have a graduate degree. And grad school is very different than undergrad in the sense that it really provides you an avenue to apply what you've learned and really expand to like what the real world application of the things are that we're learning. So one of the biggest reasons, one of the biggest factors if you want to go into grad school, aside from potential job prospects, is your curiosity to do research.
Another question in the chat is, what drew you to immunology?
So I went through my undergrad not really knowing what I wanted to do. What really drew me to immunology was the course in immunology that I took. So sort of seeing how our immune system can respond to pathogens and how we're protected. I just found it very fascinating. And also it seemed more multidisciplinary because I did biology first and everything was very monotone. But immunology seemed to thread together everything. And I found that one of the most exciting parts is that I can apply so many different disciplines to a single field. Also, nowadays, immunology is blowing up. It's like one of the number one sort of factors in a lot of the therapeutics we design, a lot of the work that we do all stems from immuno.
Another question we have in the chat is, can a baby get affected by drinking milk from a mother with some sort of virus?
I'm not sure if milk transmission of virus is well documented for things like coronavirus, but what we do know is that antibodies from a mother can be transferred to a baby through breast milk. So a couple of recent studies have shown that moms that are breastfeeding who've been vaccinated with these coronavirus vaccines, they can actually pass those antibodies to their child through breast milk. So protection can be transferred. In terms of infection, I think it's a lot rarer to happen through breast milk.
If anyone else has any questions, you can also unmute yourself and ask or type it in the chat and I'll speak it out loud. But in the meanwhile, I have many more questions.
Yeah.
So given the current pandemic, do you see any big changes that are going to happen to the field of immunology or like currently happening?
Yeah, the appreciation for infectious disease research has blown up. Before this pandemic really hit, obviously it's been a big issue, but in our society, like in North America, biggest focus has been on things like cancer, cardiovascular disease, and in a way, we let our guard down with this emerging pandemic, and it's really revitalized infectious disease research. And if you're tuning in from Canada, Canada's vaccine manufacturing capacity has been pretty much non-existent. But now that this has happened, it's really put onto the map. We need homegrown developmental facilities. And I think we're going to see a huge uprising or in the development of homegrown vaccines, homegrown therapeutics, biotech.
Is there something that you wish like the general public knew more about immunology? It's like under talked about.
Yeah, so it really comes down to like if people were more, the vaccines have been a big thing in the field right now. And one of the biggest issues is when people don't have all the proper information, it's really easy to become hesitant to accept to take something. So what to some people think is so rapidly rushed. So I think understanding the field of immunology, even at a basic level of how vaccines work, for example, how they've provided such help throughout the centuries for humanity, I think would be a major thing that people should really consider or should be considered.
Another question in the chat. How are the job prospects in immunology? What are a few career options?
Oh, it's crazy expanding. So in terms of job prospects, it really depends what field of immunology that you go into. Cancer immunotherapies right now, or treatments for cancer, have essentially highly focused on immunological aspects, utilizing T cells to target cancers to kill them, use of antibodies. Like you'll hear things in the news about anti-PD-1, anti-CTLA-4. It really comes down to a lot of cancer therapies now harness our immune system to protect us. In terms of job prospects, it can go from anything ranging from doing a PhD or a graduate studies in immunology and becoming a clinical immunologist, to working at biotech companies and developing new vaccines or developing new therapeutics. I think it's a very endless field in terms of prospects, and they're continuously growing.
Another question in the chat is McMaster University good for gaining research experience during undergrad?
Yeah, it's a bit of a subjective question. Your experiences in undergrad will depend on how much you reach out to Profs. Mac has a huge slew of Profs that are well funded. So there's a lot of opportunities, but I think that's the same case at other universities too. You just have to be really adamant in finding the position. It's always going to be competitive regardless of what university you're at.
Do you have any tips for how to reach out to Profs or get that experience?
Yeah, it's tough. The biggest thing for me when I was doing it is I tried to email as many Profs as I could. And I, send follow-up emails continuously. It's essentially like pestering them because the ones who pester them are the ones who are really interested in the research. Sometimes people send one-off emails, and a lot of Profs I've talked to here, they just ignore it. They're like, whatever. But if they're getting multiple emails from the same person, they're like, okay, maybe this person's interested in this. So be adamant and continue to ask until you get in.
Good tips. Another question in the chat was, what are your thoughts on the speed up phase testing of COVID vaccines, since vaccines tend to take 15-ish years to develop, but we develop them in about 10-ish months?
Yeah, so the reason, this is a question that's always come up when people have asked me, the reason it takes so long to develop a vaccine is because you need populations to test it in, right? So TB, for example, sure, about 10 million people a year get infected, but it's hard to find and pinpoint those individuals. So being able to get a large enough cohort of people to vaccinate and to test efficacy is hard, because it's not like the pandemic right now when you go outside and one in whatever number of people are getting infected. So the reason these vaccines have been so rapidly developed and tested is, in a sad way of saying it, is there's a lot of people getting sick. So you have a really strong statistical power to test the efficacy of a vaccine. It's not that it takes 15 years to come up with an idea of a vaccine. It takes 15 years because there's a whole lot of requirements to test efficacy. And sometimes we don't have the statistical power to test these things because not a lot of people get sick from for certain diseases that we're trying to develop vaccines against.
And going back to the research and emailing profs, do you recommend constant cold emails?
Can you elaborate on what a cold email is? Like in terms of reaching out for profs,
sure.
I like emailing them, like when I used to apply for my thesis position, just a bit more detail. I wouldn't just email, be like, hey, do you have a position in the lab for me to work in? I went and read some of the papers that lab publishes to get an idea of what they do. And I'd be like, hey, I was reading about your lab and I saw that you've done this research and you've done this recently, and I'm really interested in that. So I'm just wondering, is there any prospects for a potential thesis student? And if they don't respond to that, you follow up with like, sorry to bother you, blah, blah, blah. I was wondering if you saw my last email. And generally they bite on the second one.
So is there like
but
a
show
limit
interest in their research. Oh, like a limit of how many? I don't think so. It depends how much you love that research that you're going after, right? But the biggest thing is have interest in that lab's research and read about what they've done in the recent years, because that shows that prof that you really are interested in what they do.
Another question in the chat is, are there equally women and men in the field, or do you think there's inequality?
That's a good question. I can speak from the center that I work in, and it's very equal, I find. But outside, obviously, there are issues of inequality when it comes to certain potential positions, but not much that I can comment on because I've been pretty much essentially Mac homegrown, and I've really been focusing on the academic grad school side of research. But here, there's equality, of course. I can't speak about industry because I haven't worked in industry yet. So I don't want to speak about something that I don't know too much about.
Sort of a personal question. If you could make one revolutionary change to the industry of immunology, what would you want it to be?
I think the biggest thing is advocacy towards the general public. More money needs to be spent and more time is needed to educate the general public, because without truly understanding why we take the drugs we take or why we take the vaccines we take, just fuels hesitancy. So I think public outreach is something that should be really pushed towards.
Another question has popped up in the chat. Do you have any advice you would tell yourself back in high school?
Yeah, don't be worried about not knowing what you want to do. I personally didn't know what I wanted. I knew I kind of liked biology, but a lot of first year and second year programs are very general. Like when I took Honours Bio at Mac, I started in the life sciences program here. So it's very general. You take psychology, chemistry, mathematics, bio, and you have a couple of electives. So, see what you have a bit of interest in, but when you go into your first year, almost all first years are very general. So you can get a feel of what you like or you don't like. I knew I didn't like biology when I first started because I thought it was pretty boring and very monotone. So, I was obviously in the Honours Bio program. So I was trying different things in biology. I tried genetics, I tried cell biology, and then I tried immunology, and that's what I liked. Don't feel like you're pressured to be going into something, right? Try it all out. That's what the whole point of your first and second year, right, is at university.
Building off of the whole university chain, there's more questions in the chat. I'll go to them.
Yeah.
Is there a lot of math in immunology?
I personally am not the best at math. The main math that it's very basic. The math that we do is extremely basic. There are some chemical applications like understanding concentrations and stuff, but it's very just fundamental stuff.
How is the workload in first year life science program?
Uh, it's so the workload in comparison to high school is obviously a lot more, but the biggest change, when I look back, is you're just a number versus like when you're in high school, the profs know your name, et cetera, et cetera. In first year, you're just a number among a sea of other first years, right? So there isn't a person to hold your hand or tell you have to go study for this or do this. It's a very sink or swim in the sense that here are your courses, here's what you need to know. It's very self-directed. No one's going to be pestering you to study or to do it. So a lot of the issues people have in first year is that sort of transition from, the very structured, here's your homework, here's what you have to do, versus here are all these courses, all the midterms are on the same day, and you got to go study and organize your own time. So time management becomes a big thing and really self-motivation to study.
Yeah, I'm also first year at university right now, so I think I'll just build off a little bit. Expect your grades to drop a bit because like you're surrounded by all these people who got really good grades in high school, so it's going to be more competitive and it is going to go down a little bit. Just be prepared for that.
Yeah, big time, big time.
What advice would you give to students transitioning from high school to universities?
Yeah, the biggest thing is stay on top of your work. When it becomes so self-directed, it's really easy to chill and not stay on top of what you're doing. So as soon as you start falling behind, it just piles on top of each other because, just because like in high school, for my high school at least, a lot of my courses talk to each other. So they try not to overlap things. But when you're in high school, it's not like biology talks to chemistry. They don't care. They'll just pile everything on top of each other, right? It doesn't matter what else is going on. So manage your time and stay on top of the work.
Any tips with maintaining mental health in university?
Yeah, that's an excellent question. So, especially during a pandemic like this, mental health has become more and more of an important thing. Don't be afraid to get help. Universities have so many options of therapy or counseling or just having, groups you can talk to. There's nothing wrong with it. Take advantage of the things that we have offered to you in university. It's worth it. It helps.
Could you elaborate on your first time working with a prof? What kind of responsibilities did you have?
Yeah, so that's a very subjective question. It depends on what lab you're working in. So I'll see some first year, second year, third years work in some labs here where they're essentially on they're on menial tasks like fix this, fill this, do this. Whereas some labs have students go into like an actual project. So do your research beforehand and talk to the people in the lab if you get an interview to know what the lab environment is like. Generally, when labs are really big, incoming undergrads most of the time get sort of menial task stuff to do, like go fill this, go do that. Whereas if it's a bit of more of a tight nip or a tight lab, you might actually have the opportunity to work on a project yourself. So it's very heterogeneous. It really depends on the lab that you're looking at. So do your research in and out, not only of the prof, but also the lab itself. And usually lab websites are a great indicator of how many undergrads there are, what they do. So do your research.
Just building off of the whole research as a first year, if you're working in a lab, if you mess something up, what happens?
The biggest thing is you just gotta tell them what the mess up was, right? It doesn't things happen. I mess up things all the time in a lab. It's just part of the natural work that we do. Something messes up, you be like, hey, screwed this up. What can we do to fix it? Or provide a suggestion like, hey, I screwed this up, but this is how I know I'm gonna fix it. So always be prepared to answer why it happened and how you can fix that issue. It's always bad practice to be like, hey, I messed this up. Sorry. Have a reason of why something happened and how you can fix it.
What would you consider is your greatest achievement so far? in your career studies?
I think a lot of it really stems from the vaccine work that we've been able to do in terms of our academic achievements. I think one of my favorite achievements, really, I taught third year immunology here, and I just absolutely loved being able to teach others and, seeing them go from this state of, similar to how I was, I'm not too sure if I liked immunology, to really seeing that passion get lighted underneath these people. So one of my biggest achievements really comes from the teachings that I did here.
A question in the chat, could you try to contact profs at a different university?
For graduate school, absolutely. If you want to work in undergrad labs, yeah, for sure. As long as you have the means of going to that university and carrying out the work, there's nothing stopping you from doing that, right? If anything, it's better to get yourself out there, outside of your comfort zone, build that resume, get those reference letters. And that really starts from your first year. I didn't do much of that in my first year because I didn't know what to do. But one of the biggest advice to sort of loop back to the first question asked is be proactive. Because every year that goes by, you can add another notch into your resume belt, into your reference belt. That'll become just that much more useful down the road to you.
And going back to the screwing up, another question was saying on any tips on building resilience, since there's a lot of ups and downs when studying.
I think that's why we call it research, right? Because you constantly have to go back and redo things. It's not just search. So even to this day, I'll screw up things in the lab. I'll be like, oh God, why am I here? But that's the whole, over time you become used to it in the sense that you accept that it's part of the process. But use screw ups or potential bad results of experiments or whatever you do as learning opportunities to go back and improve on yourself, develop new experiments, for example, if it's research that's failing, or if you screw up, use it as an opportunity to know why you screwed up, and you can use that to teach others. So like, I know things that I've done that I've messed up in the lab, and now I can tell the grad students that I work with what they can look out for so they don't do it either. It's a learning opportunity.
Do you have any plans for your future? Like, what do you plan on doing after completing your PhD and so on?
So my plan, I'm hoping, is to do either industrial research for vaccines. Most likely that's going to be in the US, or potentially working for agencies like Health Canada and being part of sort of National Research Council. And I still want to stay in the sciences and do hands-on work with research. So I have a goal in mind, but the first step is really to get through this postdoc.
What was your favorite part of undergrad?
Yeah, that's a good one. Really, it's kind of a generic answer, but it's really the ability to make these new friends and new groups and sort of live that undergrad life around in the city. Like I'm in Hamilton right now, so like, going out and jumping around places with your new group of friends. It's hard right now with COVID going on, and it really sucks for incoming first years. I mean, for you said you're in first year, right? It must suck to just be at home looking at lectures online.
Yeah.
So it really makes you appreciate how different things are when you have the opportunity to go out and hang out with friends and just, bar hop, for example, or do anything like that. That's definitely one of the best ones is the interactions with your buddies. And it's kind of weird to say, but like you guys all have exams coming up. So everyone just comes over and you're just all dying in the same room studying for the exam. It was kind of fun just to hang out with everyone.
Any tips for building friends or connections, especially with online university?
How would I route that question to you? Because you're first year.
Sure. Yeah. Well, I go to UBC, so I'm in British Columbia right now. And I think like the universities, they're really trying to get you engaged. They moved everything online, but like they still have our like clubs days where you meet all these different clubs and you can join them. So I definitely recommend trying to do that. And there's also all these Instagram accounts where you can post a little quick introduction to yourself and then people will contact you based on that. So basically it's just about getting out there, but also I think even if you don't make any friends, it's gonna be okay. I think it'll be a little easier once things start to go back in person, hopefully.
It will. It's coming.
Do you know,
This COVID stuff won't last forever.
as an immunologist, you have like a predicted time when everything's gonna go back to normal?
These first generation vaccines are doing phenomenal things. I know certain universities like McGill is 100% in person next year, but like U of T, I believe is online from what I've heard. And I think a couple of other Ontario schools are going to be like 70% in person, 30% online. It's just, it's too, yeah, like Ryan said in chat, I think Western is doing that 70-30 split, but it's, no one knows. Now with these variants coming out, who really knows how these things advance? It's hard to say. We didn't think about variants when we first started doing this research because usually variants take time to develop. But COVID itself is huge. It's spread like crazy. So I think if vaccine rollouts continue and continue to accelerate, just like that pandemic flus we've had in the past, it'll just become seasonal, just like everything else. It just takes time.
So I'm just trying to think of a question or.
No, it's okay. You can always email me if you guys have questions or anything.
Does anyone else here in the meeting have any more questions? Ryan is asking about your email, and there's another.
Yeah, here. Yeah, feel free, guys.
And there's another question in the chat. I've heard that being in the research field requires jumping from job to job. Is this true?
Excellent question. Depends on what you want to do. If once you go through your PhD, it's always sort of, it's not required, but it's sort of in a tradition where you go and do a postdoc. Usually your postdoc is done in another university or in the same university. The real answer, I guess the answer to that is depends on what you want to do. I know people here who've stayed here from the start of their undergrad and are profs here. And I know some people who expanded outwards and moved to companies in the US. It really depends on what you want to do. In terms of job to job jumping, it comes down to what you personally want to do. I know people who've been in the same company position for years now versus others who get sort of bored and want to move up. To follow that also, it depends what type of degree you have. So there's a lot, sometimes there's a ceiling when it comes to if you have a master's or a bachelor's only, you can only go so high in certain companies. But once you've expanded into the PhD field, you've opened up a lot of opportunities in terms of higher level jobs like scientist positions and etc. But it all comes down to what you want to do. Nothing is static. Never think that you're just stuck on one road.
Any last minute questions from anyone? Do you know if there are good scholarships for international students?
Oh, that's a good question. You have to look on, every university has their own section for scholarships and bursaries. Go check those out because every university will have their own in-school scholarships and bursaries, and then I'm sure there's international ones. It's been a while since I've applied for these things, so go and check the university's websites.
Last call for any questions, or we can just end it here. We're pretty much at the hour mark anyway.
Perfect. Well, I really appreciate you having me. It's a great incentive or initiative you guys are doing. So keep it up.
Thank you. think that's about it. Thank you so much for being here today. Oh, yeah.
Yeah, thanks so much. I appreciate it. All right. Yeah, anytime. If you have any questions, just shoot me an email. Thanks, guys.